So I think we can move on with our next speaker. I guess he's in the backstage. So our next speaker is Luca Maragliano. He's
Hello.
going to talk about molecular dynamics simulation of a tight junction paracellular channel. Luca, please.
Yes.
Go ahead.
Okay, so let me, do you see my slides?
Yes, great.
Okay. Okay, so good afternoon, everybody. First, let me thank the organizers and the committee for giving me this opportunity and for the efforts in setting up such an interesting workshop. In also given the difficulties of this period. Now, my talk will be about a class of ion channels that are quite different from the most commonly discussed ion channels that regulate traffic across the cellular membrane. And so I need a little bit of a biological introduction to those systems. So I will be showing our work on tight junctions channels. And tight junctions are quite complex molecular architectures that bind together epithelial or endothelial cells in biological barriers, where they regulate the paracellular transport, so the transport in the restricted space between the cells. Now, biological barriers are throughout the body. Essentially, they separate distinct compartments. For example, the blood vessels and the organs where nutrients have to be delivered, and the transfer of nutrients can occur across the endothelial cells, and this would be the transcellular transport, or across the paracellular pathway, and this is, as said, regulated by the tight junction architectures. Now, these barriers, and tight junction in particular, are major targets in drug delivery, and it's particularly so for the tight junctions, because along this pathway hydrophilic molecules can be made pass. Now, tight junctions, as we can see in this figure here on the right, I hope you see my arrow. If we look at the this is a picture taken from Frick's fracture electron microscopy. Tight junctions are organized in strands. These lines that you see here, these are strands of tight junctions, and they are usually represented schematically as the figure that you see here below. So strands from one membrane of one cell are in contact with strands from a membrane of the facing cells. These contacts are in fact the tight junctions, or also they are also called sometimes kissing point. We see here another figure from electron microscopy. Now, these tight junctions are made of proteins, adhesion proteins, and the most important class of proteins among them are the claudins. There are many claudins in all the organisms, and they are expressed differently in the different tissues, so in the different barriers. I will be talking about Claudin 15, that is in the intestinal barrier, and about Claudin 5 at the end, which is in the blood-brain barrier. So depending on the tissue where they are expressed, claudins can work as channels, as selective channels, in fact, or as barriers. And here on the bottom right, I'm showing work from the group of Turner at the University of Chicago with Chris Weber, where they were able to insert a pipette into a tight junction and measure the channel activity of the Claudin 2 tight junction. This was impressive electrophysiological work. Now, from the structural point of view, little is known about Claudin proteins. The first Claudin that was solved from X-ray was Claudin 15 in 2014, which is in the intestinal barrier. It's a channel-forming Claudin, and it is cation selective. So it is made of 4 transmembrane helices, and it has 2 extracellular loops, there is a gap, a small gap in one of the loops. And it's important also to note that in this extracellular loop, there are negatively charged residues that will have an important role in what I will tell in a moment. So the same group that solved the structure of the protomer of the single protein actually proposed a structure for an architecture of Claudin 15 strands forming pores. Now, this model, which is now referred to as the Suzuki model, was made so based only on the crystal structure of the single protein and the crystal packing, but it was made consistent with images from freeze fracture electron microscopy and from cross-linking experiment. But the pairing of the two layers from adjacent cells was made just by making closer the layers of the crystal structure of the protomer. So the limitations of this model were that because clashes were created, so they had to cancel some loops, and so there were no native protein-protein contacts, and most of all, no interaction with the membrane. So this is where we came in with the aim of testing and maybe refine this model for a Claudin 15 based tight junction pore using molecular dynamics simulations. So we extracted the minimum number of protomers that can form a pore, which is 4. We modeled the missing loops from the Suzuki model, and we embedded the two different dimers in two membranes, and of course we solvated in water. So this is the structure that we made for a single pore, and we also investigated a double pore to explore different conformations of different possible conformations of the loops. But I will be showing results mostly for the single pore. So this is how the single pore structure looks like. Just to let you appreciate it in 3 dimensions. So a nice feature is that the aspartic the aspartic acid 55, the negatively charged residues that I mentioned before, when the protomers are brought together, they actually stay at the center of the pore. So there is the pore as a beta barrel structure, and the negatively charged residues are in the middle of it. And there are also other negatively charged residues that line the surface, the inner surface of the pore. If we look at the interior of the pore, just by rotating this figure by this structure by 90 degrees, and if we delete the two protomers that would be here in front of you, you can see how the other negatively charged residues are lined. So we first did standard MD simulations just to evaluate the stability of this structure. So the tetramer is stable in the double membrane. The single protomers maintain their conformations, and interestingly, the tetramer is held together by interactions among clusters of residues that are known to be conserved among claudins, also of different families. So there are hydrophobic clusters among trans dimers, so dimers from opposing cells, and then there are hydrogen bonds among cysteine residues in cis dimers, so dimers in the same membrane. If we look at the sides of the pore, so the maximum constriction side has a radius of about 3 angstrom, and it is at the level of the ring of aspartic acids. So if we look at at how this maximal constriction point, so the minimal radius point, evolves along the simulation, we observe some fluctuations at the beginning of our trajectory, but then it stabilizes. Here I'm showing different structures corresponding to these different points in the trajectory, and then it stabilizes at a radius of about so a diameter of between 5 and 6 angstrom. So this is a structure corresponding to the stabilized region. So we were quite happy with this for the structure of the pore, but what about the function? So I've mentioned that Claudin 15 are cation selective. Here I'm just showing results from experimental papers by other groups where they show that the permeability for sodium is higher than the permeability for chloride for these systems, and that this cation selectivity is actually due to the presence of the negatively charged residues at the center of the pore. Because as you can see here from dilution potential measurements, when the aspartate 55 is mutated to a lysine, then the dilution potential is actually inverted. So we asked ourselves if the structure that we had refined was in fact recapitulating these features, so if it is also cation selective. We used the standard method, umbrella sampling method at first, so much more simple method than those described by Christoph just now. And so we used umbrella sampling for ion translocation through the pore from what is called the apical side to the basal side, and we investigated sodium, potassium, and chloride, and we obtained this profile. So for cations, for sodium and potassium, we have an attractive minimum of -4 kcal per mole, while for chloride we obtain a barrier of about 8 kcal per mole, and those features actually are in correspondence with the ring of D55 of the aspartic acids that I have mentioned already. And if we look at how sodium ion is bound at this binding site, at the minimum of the PMF profile, and we look at its coordination along the umbrella sampling windows, we see that there is alternate coordination from water molecules and protein atoms, and specifically the oxygens from the aspartic 55. So then we look also at the kinetics of the ion going through the channels, and we use the Voronoi-based milestoning method, which is a version of the original milestoning designed by Ron Elber. The Voronoi method was devised by Eric van den Eijden and Maddalena Venturoli in 2009, and it amounts at restricting the trajectory in windows, in cells, whose walls are identified with the milestones. And this restriction can actually be, this confinement can be realized by hard reflections at the boundaries. This was the original recipe by Eric and Maddalena, or as we with Eric and Benoit proposed at the same time, also with bouncing potentials that actually are activated only when the trajectory crosses the boundary. So with milestoning, again, we studied the permeation of sodium, potassium, and chloride. And so first, from milestoning, we can get the free energy of being inside each of the cells that are along the channel pore, and so we get the free energy. The free energy profile is practically the same of the one that we got from umbrella sampling, but we can get also the mean first passage times of going from the most external milestone, which is at the apical side, to the most internal milestone, which is at the basal side. And so the transition times that we obtain, confirming again cation selectivity, are of about 1.1, 1.6 for microseconds for cations, and 43 microseconds for chloride. So this is where the main results that we have so far. Obviously, we were not the only group working on these systems. After the publications of the Suzuki model, actually different groups have been working on these systems. I would like to mention in particular the work of Shik and Anja, that has been from Syracuse University, that has been simulating different Claudins in different pore conformations, and also the work of Fatime Khalili Araghi, at the University of Chicago that many of you know very well. They did nice work on Claudin 15, and they get similar results to ours. Now, the next step that we are pursuing now is moving to the tight junctions of the blood-brain barrier with the goal of designing devices that can actually transiently open these tight junctions without disrupting them. Now, the claudins that are expressed in the blood-brain barrier are the claudin 5. There is no experimental structure for claudin 5, but it has a similarity of 40% with claudin 15. So we build an homology model, and we equilibrate it in the membrane. And now the question is, the blood-brain barrier is much more restrictive than the intestinal barrier. So it's much more selective. So the idea is whether the claudins are organized in a pore that is like the Suzuki one, but the dynamics of the extracellular loops is different, and so occasionally they close the pore, or if they are arranged in completely different dimers, and when the cells are together, these dimers give rise to different pore conformations. So we are actually exploring both hypotheses. And so here I'm showing a Suzuki-like pore for claudin 5, and this is, so we started doing simulations on that, but I mean, from the first configurations that we obtain, we don't see remarkably different pore opening with respect to claudin 15, but it's also important to consider that the residues that line the interior of the pore in claudin 5 now are different with respect to those that were lining the interior of claudin 15. In particular, in claudin 5, there is no ring of charged residues at the constriction site. There are other positively charged residues, but they are more distributed, and they are in regions of the pore that are wider. And so we are also looking at peptides in order to transiently open the tight junction. And we extracted peptides from a toxin that has been shown to have to actually disrupt the blood-brain barrier tight junction, so to target claudin 5. We extracted a set of peptides from the minimal interacting surface of the claudin with this toxin. And so we remodel, and we actually here I'm showing the 3 of them, but we model them separately. And this part of the work is in collaboration with the experimental colleagues at the Italian Institute of Technology, where they have set up what is called the transwell system. They can grow barrier cells on a device that separates the 2 compartments, and then measure the what is called the transepithelial electrical resistance in the different conditions, and in particular, it will be done in presence or in absence of these peptides. Now, these are just control simulations. So to summarize, I've showed you our refined model that we obtained via Olatom MD of a claudin 15-based tight-junction paracellular pore. It has a minimal pore size of 6 angstrom. This result is consistent with estimates that were done from experimental data before our work. And we obtained from free energy calculation that is in fact cation selective pore, and this selectivity, the selectivity feature, are in correspondence with the aspartic 55, as was hypothesized from experiments before. And then to conclude, I would like to thank the people working also on this project, Fabio Benfenati, the director of the Ensign Center at the Italian Institute of Technology, the simulation group that is working with me there, Giulio Alberini. This work was the work of his PhD. Now Alessandro Berselli has taken his part, and then the guys working on the experiments, Mattia, Valentina, and Eleonora. And with this, I thank you for your attention.
Okay, Luca, thank you very much for your talk. There are questions on the chat. The first one comes from Mark Sansom. He's asking, what is the assumed ionization state of the acidic residues in the narrowest region of the pore? Will they all be fully deprotonated?
No, they are actually, yes, sorry, they are deprotonated. They are charged, and they are charged because, in fact, the water can access the pore, so they are usually solvated, and so we used it as charge. As also the other simulations were done with charged aspartic acids. Simulations from other groups.
Yeah, there are a couple of questions. We are running short of time, so I will make them together. So the first one is there any selectivity among cations, divalent cations? This is the first one. The second one is for Armagan Koser, asking, how do the two parts of the tight junction performing membrane proteins align to each other in opposing cells?
So the first one, we didn't look at divalent cations. There's no as far as I know, there's no experimental evidence for divalent cations going through Claudin 15. There are other Claudins that are selective for divalent cations. But other groups have investigated monovalent cations, but of different sides. So they were able, in fact, to see the group of Fatimi Khalili, in fact, from simulations. They investigated Mea and Tia cations, and they, in fact, saw that the size, the maximum size for the ions was 3 angstrom. The second one, sorry, I didn't get it. Could you repeat?
So I asked it again. How do the two parts of the tight junction performing membrane proteins align to each other in opposing cells? So it's asking how the two parts they face each other, right?
Yes, I don't know how the process is formed actually.
Okay, so there is another really fast one.
I don't know. I think they're brought there by other proteins, but I don't know exactly how. So there are other proteins that constitute the scaffold of the internal scaffold of the tight junctions, but I don't know the details of how this is formed.
Okay, really last one. Has the membrane any active involvement in tight junction function? And if so, do you model them?
The membrane itself?
Yep, the membrane itself, yes.
We model just as the environment, keeping it so holding together the transmembrane helices of the Claudins, and we didn't see any particular for the size of the system that we simulate. We didn't see any particular curvature in the or deformation of the membrane. There are actually other models of tight junctions that are based on joining of the lipids, but that's a different hypothesis, and of course it doesn't take into account proteins, and so we are working on the proteins, on the protein hypothesis, for which there is solid experimental basis because they have been reconstructing these barriers by protein themselves.
Okay, so Luca, thank you again. There is a further question that maybe can be asked on the private chat. You should be in touch, sure, one with the other, so maybe the question can be posed over there.
Okay.
So we close the session here. We have a very short break, actually 8 minutes. So we are going to restart at 16:40 sharp. So thank you for being online.